Good day learners, I am Jello Helberio, and for today's video, we will be discussing 3 topics that I have chosen on the subject microparasitology. Our first topic will be all about biotechnology. Are you familiar with the products that are lactose-free? As we all know, lactose is a natural sugar found in milk. But why should we get rid of lactose? It's because many people have a peculiar intolerance towards this natural sugar. However, lactose-free products are not an option until a few years back then. It's all possible now due to the advancements in the field of biotechnology. What exactly is biotechnology? According to the Organization of Economic Cooperation and Development, it is the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. Now, let's take an example. Do you know how bread is made? The dough is added with the organisms like yeast, which helps in releasing certain chemicals. The reactions ultimately gives us the soft and fluffy loaf bread. On baking here, we use yeast organism to obtain the product on our own interest. So, do we mean that bread making is a part of biotechnology? Certainly yes. Processes like fermentation are considered as the part of biotechnology as they include the utilization of organisms. Thus, obtaining products for human benefit like food items, medicine, hybrid crops, and many more are possible due to biotechnology. Now, before we proceed to our next topic, let's give another example which turns something more amazing with the help of biotechnology. Are you familiar with golden rice? Is this rice made of gold as the name implies? No, absolutely not. Then why is it called golden rice? Well, the rice we usually see is whitish in color. However, the golden rice we are talking about is the modified version, which is yellowish in color, hence it is called golden rice. But what makes this rice yellowish and so important? It's because of the presence of beta-carotene and the help of GMO. And what does it mean by beta-carotene? It is a red-orange colored pigment which gets converted to vitamin A in the human body that helps the development and maintenance of good vision. Thus, this was done by exploiting a biological presence, making it a classic example of biotechnological achievements. Now, moving forward to our next topic, let's discuss about soil microbiology. When we say soil microbiology, it is the study of microorganisms in soil, their functions, and how they affect soil properties. It is believed that between 2 and 4 billion years ago, the first ancient bacteria and microorganisms came about on Earth's ocean. These bacteria could fix nitrogen, in time multiplied, and as a result release oxygen into the atmosphere. This led to more advanced microorganisms, which are important because they affect soil structure and fertility. Soil microorganisms can be classified as bacteria, actinomycetes, fungi, algae, and protozoa. Each of these groups has characteristics that define them and their functions in soil. So first we have here is bacteria and archaea. They are considered as the smallest organisms in soil apart from viruses and are prokaryotic. They are the most abundant microorganisms in the soil and serve many important purposes including nitrogen fixation. Some bacteria can colonize minerals in the soil and help influence weathering and the breaking down of these minerals. The overall composition of the soil can determine the amount of bacteria growing in the soil. The more minerals that are found in area can result in a higher abundance of bacteria. These bacteria will also form aggregates which increases the overall health of the soil. Second is what we call actinomycete soil microorganisms. They are a type of bacteria but they share some characteristics with fungi that are most likely a result of convergent evolution due to a common habitat and lifestyle. Next is fungi. We commonly see this around as they are really abundant in soil, but bacteria are more abundant. Fungi are important in the soil as food sources for others, larger organisms, pathogens, beneficial symbiotic relationship with plants or other organisms, and soil health. Another one we have here is algae. One unique character it has is that it can make their own nutrients through photosynthesis. We all know that photosynthesis converts light energy to chemical energy that can be stored as nutrients. For algae to grow, they must be exposed to light because photosynthesis requires light. So algae are typically distributed evenly whenever sunlight and moderate moisture is available. Now lastly is protozoa. They are eukaryotic organisms that were some of the first microorganisms to reproduce sexually, a significant evolutionary step from duplication of spores like those that many other soil microorganisms depend on. Moving forward to our last topic, let's tackle classification of microorganisms. Life on Earth is famous for its diversity. Throughout the world, we can find many millions of different forms of life. Biology classification helps identify each form according to common properties, using a set of rules and an estimate as to how closely related it is to a common ancestor in a way to create an order. The first, largest, and most inclusive group under which organisms are classified is called a domain and has three subgroups: bacteria, archaea, and eukarya. The domain was proposed by the microbiologist and physicist Carl Woese in 1978 and is based on identifying similarities in ribosomal RNA sequences of microorganisms. The second largest group is called a kingdom. Five major kingdoms have been described and include prokaryota, archaea and bacteria, protactista, protozoa and algae, fungi, plantae, and animalia. A kingdom is further split into phylum or division, class, order, family, genus, and species, which is the smallest group. The science of classifying organisms is called taxonomy, and the groups making up the classification hierarchy are called taxa. Taxonomy consists of classifying new organisms or reclassifying existing ones. Taxonomy consists of classifying new organisms or reclassifying existing ones. Microorganisms are scientifically recognized using a binomial nomenclature using two words that refer to the genus and the species. Methods of classifications are constantly changing. Classification of microorganisms has been largely aided by studies of fossils and recently by DNA sequencing. More importantly, the most widely employed methods for classifying microbes are morphological characteristics, differential staining, biochemical testing, DNA fingerprinting or DNA-based composition, polymerase chain reaction, and DNA chips. So that's it. I hope you learned something from this discussion and thank you for watching.